Hello AP Biology students, we're going to go over the 8th section in Unit 5. And in this section, we're going to talk about something called non-Mendelian genetics. It's important to understand the difference between Mendelian and non-Mendelian genetics after this section. Basically with Mendelian genetics, we're looking at an inheritance pattern where a single gene is going to control a trait, and it's usually only controlled by a dominant and recessive allele. Within non-Mendelian genetics, it's not going to follow those simple rules, and this means the outcomes are going to be a lot more difficult to predict. Our first type of non-Mendelian genetics that we're going to talk about is something called multiple allele traits. And just like the name implies, this trait is going to be controlled by several different alleles. This is compared with simple Mendelian genetics where there were basically only two alleles, a dominant or recessive version. In multiple allele traits, there are more than two, and how those alleles interact is going to determine the phenotype of the organism. Most human genes are controlled by multiple alleles. As you can see in our chart, we have our simple Mendelian genetics where there are only two alleles, but in multiple allele traits, there are many alleles. The example I'm going to use for multiple allele traits is like when there are multiple alleles for fur color in rabbits. How those alleles are going to interact is going to determine what the phenotype of the organism is. And since there are multiple alleles, there are going to be more versions than just two phenotypes. Our second type of non-Mendelian genetics is something called incomplete dominance. Remember, with normal Mendelian genetics, we only have two phenotypes. We have the dominant phenotype and the recessive phenotype. In incomplete dominance, the heterozygous form is going to have a distinct and different phenotype than the two homozygous phenotypes. You can see in this example, the phenotype is going to be a mix of the two alleles. In this case, it's green, which is a combination of yellow and blue. An example of incomplete dominance is like snapdragon flowers, where we have a red version and a white version, but the middle or the heterozygous version here is a mix of the red and white, which makes pink. Another closely related type of non-Mendelian genetics is called codominance. In this type of inheritance, the heterozygous form is going to show both of the phenotypes. You can see here we have a yellow and blue phenotype, but the heterozygous form here is going to show both yellow and blue. Example of this in real life is called a roan cow, where we have pure red cows, pure white cows, but the roan or the heterozygous form shows both red and white. Our next type of non-Mendelian genetics is called polygenic inheritance. This is where several genes are going to contribute to a single phenotype. Up until this point, we've been really looking at one locus and determining how that locus affects one phenotype. In this type of inheritance, there will be many genes that affect one phenotype. Again, most of human genetics follows this type of inheritance where there are many genes that will affect that one type of phenotype. And since there are many genes that are going to affect one phenotype, you can see that the end result's going to have a bell curve effect of phenotypes. Human height is an example of this where there are many genes that will affect the one phenotype of how tall a human is. And as you can see in the example here, both men and women have a bell curve of data that shows how tall a person can be on average. Our next type of non-Mendelian genetics is called variable expressivity. This type of inheritance refers to when there is a range of outcomes for a single genotype. Yes, this kind of goes against what we learned in Mendelian genetics, where if you have like a dominant or recessive trait, you're going to have that phenotype. Marfan syndrome is an example of this variable expressivity, where just because you have the genes or mutations that cause this syndrome, there's going to be a range of outcomes of how severe it is in a person. Our next type of non-Mendelian genetics is called incomplete penetrance. This is where individuals who have a genotype might not produce the phenotype. Example of this is the BRCA1 gene or the BRCA1 gene that mutates and can produce breast cancer. Not everyone that gets a mutation in this BRCA1 gene will develop breast cancer, so it's an incomplete penetrance since not everyone that has a specific genotype will produce the phenotype. Our next type of non-Mendelian genetics is called pleiotropy. This is where a mutation in one gene will have an effect on many different phenotypes. For example, people that have a mutation in a pigmentation gene are classified as being albino since one gene is affecting many different phenotypes. Our last type of non-Mendelian genetics is called epistasis. This is where there is an interaction between two genes where one gene kind of has control over another gene. I like to think of this first gene as being like an on-off switch for the second gene. And that interaction is going to create more than just two phenotypes in the offspring. An example of this is like the pigmentation genes we see in Labrador dogs, where if the first gene is turned on, we have either chocolate or black labs. But if that first gene is turned off, then you can see this is where we get the yellow labs. It doesn't matter what the second gene is, it's going to be yellow because the first gene is turned off. Now, I'm not going to go over how to go through each type of Punnett square with you in this TikTok, but I have made other TikToks about how to work through these types of inheritance patterns, so just check those out.